Hello everyone, I am Dr. Amit. Please do subscribe to my YouTube channel and press the bell icon for more medical content. Today we are going to deal with one of the most important and confusing part of our biochemistry series. Yes, today we are going to deal with glycolysis. So please pay attention. By the end of this video, I am sure you will never get nightmares about glycolysis or its ATP production, and you will never miss MCQ on this topic again. So let's start our today topic, glycolysis. So first let's understand the basic that glycolysis occur in cytoplasm. We have already learned about what are different cycles of biochemistry and where it occurs in the previous video. Okay, so we all know that glycolysis occur in cytoplasm. Now we'll talk about different steps of the glycolysis one by one. Okay, so glycolysis starts with glucose. Okay, so we have one molecule of glucose which is getting converted to glucose 6-phosphate, and the enzyme used over here is hexokinase or glucokinase. We all know that we use glucokinase where the concentration of glucose is high, like in liver or beta cell of pancreas, and hexokinase in other tissues. Okay, so the main work of kinase enzyme is to phosphorylate this glucose to glucose 6-phosphate, or we can also say that to add one phosphate to this glucose. Okay, so now after adding one phosphate to this glucose is getting converted to glucose 6-phosphate, and during this step we are losing one ATP. Okay, it means one ATP is used to convert glucose to glucose 6-phosphate. Okay, now this glucose 6-phosphate is getting converted to fructose 6-phosphate. Okay, after that this fructose 6-phosphate is getting converted to fructose 1,6 biphosphate. Okay, so in this step we are using the enzyme phosphofructokinase 1, and this step is very important because many MCQs are asked from this step. Okay, this is also called as rate limiting step. Okay, so most of the time MCQ asks like which is the rate limiting step of glycolysis. So converting fructose 6-phosphate to fructose 1,6 biphosphate. Okay, and which enzyme is used in rate limiting step? Phosphofructokinase 1. Okay, and in this step also we lose one ATP. Okay, so it means one ATP is used to convert fructose 6-phosphate to fructose 1,6 biphosphate. Now the next step is fructose 1,6 biphosphate is getting converted to glyceraldehyde 3-phosphate. Okay, and in this step we are using the enzyme aldolase. Okay, so we use aldolase B in liver and we use aldolase A in muscle. Okay, now the next step will be, now this glyceraldehyde 3-phosphate is getting converted to 1,3-biphosphoglycerate. Okay, so in this step, okay, we get 2 NADH. Okay, here is no loss, here is gain, so we use this 2 NADH. Okay, so later we'll talk how this NADH is getting converted into ATP. Okay, just remember that in this step we get 2 NADH. Okay, now this 1,3-biphosphoglycerate is getting converted to 3-phosphoglycerate. Okay, and in this step we are using the enzyme phosphoglycerate kinase. Okay, and in this step we are gaining. Okay, we are not losing, we are gaining 2 ATP over here. Okay, now this 3-phosphoglycerate is getting converted to 2-phosphoglycerate, then 2-phosphoglycerate into phosphoenol pyruvate. Okay, now this phosphoenol pyruvate is now getting converted to pyruvate. Okay, this is where the glycolysis end. Okay, this phosphoenol pyruvate getting converted to pyruvate, and this step in this step we use the enzyme pyruvate kinase. Okay, and in this step also we gain 2 ATP. Okay, so while overviewing this all the steps, we are losing 1 ATP over here, we are losing 1 ATP over here, but we are gaining 2 NADH over here, we are gaining 2 ATP over here, and we are gaining 2 ATP over here. So total loss of ATP is 1 and 1 here, 2, and total gain of ATP is 2 over here, 2 over here. So total 4 ATPs we gain and 2 NADH we are gaining. Okay, in this cycle. Now we'll see the net equation. Okay, so while talking about the net equation, we use one molecule of glucose, okay, and 2 ATP to produce two pyruvate molecules. Okay, so from one we'll get two pyruvate molecule. Okay, so we are getting 2 pyruvate and we are getting 2 ATP. Why 2 ATP? So total ATP we are getting 4, but we have lost 2 ATP, so 4 minus 2. So 2 ATP you are gaining, 2 NADS we are gaining from this step, okay, and 2 H plus and 2 H2O. Okay, now we'll look how we are converting this NADS into ATP and how many ATP we produce in aerobic cycle and anaerobic cycle. Okay, now this is the most dangerous part, we can say. Okay, or we can say this is where the most student get confused. Okay, because how the ATP yield under different conditions, or we can say that how NADS is getting converted to ATP, okay, in different conditions. Okay, so most of the examiners love to ask questions from this step. Okay, so now we'll read about net yield calculations. Okay, so the glycolysis can occur in aerobic conditions or anaerobic conditions, or can occur in aerobic conditions. Okay, so in anaerobic conditions there will be absence of oxygen. Okay, so when there will be absence of oxygen, so the NADS produced over here, okay, this NADS, okay, this NADS produced over here can't enter mitochondria. Okay, so if NADS can't enter mitochondria, then it will not get converted into ATP. Okay, so now this NADS will be used to reduce pyruvate to lactate. Okay, now we'll see how ATP are getting produced over here. Okay, so we know that total ATP produced is 4. Now total ATP used is 2. Okay, we all know that 2 ATP is lost and 4 ATP is getting produced. Okay, and this we can't produce NADS ATP from NADS because there is absence of oxygen. Okay, so the net gain will be 2 ATP. Okay, so whenever the examiner will ask how many ATP is produced in anaerobic conditions from glycolysis, then we will say 2. Now we'll see that in aerobic conditions. Okay, so in aerobic conditions there will be presence of oxygen. Okay, oxygen is present, so the NADS can enter mitochondria to undergo oxidative phosphorylation. Okay, so oxidative phosphorylation is a process through which NADS will get converted to ATP. Now in oxidative phosphorylations there are two gates. Okay, one is malate aspartate shuttle and the second one is glycerol 3-phosphate shuttle. Now if the NADS will enter through this step, then one NADS will produce 2.5 ATP. But if NADS will enter through glycerol 3-phosphate shuttle, then one NADS will produce 1.5 ATP. So it depends on from which gate the NADS is entering, okay, the oxidative phosphorylation. So we all know that total gross ATP we are producing is 4. The total ATP used during the cycle is 2. Okay, now the total NADS we are producing in glycolysis is 2 in this step, right? Now if these 2 NADS will enter to malate aspartate shuttle, then one NADS will produce 2.5 ATP, then 2 NADS will produce 5 ATP. If this NADS will enter the glycerol 3-phosphate, then one NADS produce 1.5, it means 2 NADS will produce 3. Now how many ATP will produce? So total 4, okay, minus 2, and if it enters the malate aspartate, then total ATP will be 7. If it enters glycerol 3-phosphate, then 5. So if the examiner will ask that how many ATP is produced from in aerobic conditions, then from malate aspartate shuttle 7, and from glycerol 3-phosphate shuttle total ATP produces 5. So this is all about glycolysis. Okay, so I hope this video will find very useful to understand glycolysis, and thank you for watching. Keep hammering your brains. One day you will shine. If you like this video, please subscribe, like and share it with your fellow medicos, and see you in the next one. Thank you.